who leads the stem cells and cancer laboratory at the Monash BDI, and Professor Paul McMurray, who's the head of surgery in the Cabrini Monash University Department of Surgery. And so it's, I'm presenting to you today sort of these many years of work we've put into building this comprehensive colorectal cancer bioresource, and to chat with you a little bit about how we're using these patient-derived organoids in our preclinical work in colorectal cancer. So as many of you will be aware, my slides want to change, there we go. Colorectal cancer is a huge health burden in Australia, with 1 in 13 Australians due to develop colorectal cancer in their lifetime, making this the 4th most commonly diagnosed and our 2nd biggest cancer killer in Australia. It is also the leading cause of death, cancer-related death for young people aged between 25 and 44 years, so no longer really considered just an old people's disease. Concerningly, we are seeing an increased incidence in this early onset colorectal cancer, and these patients often present with quite advanced disease in stage 3 or 4, where the disease is more difficult to treat. So there's an urgent need really to try and improve treatment strategies for these patients. We do know that bowel cancer is one of the most treatable cancers, with 99% of cases treated successfully if found early. However, fewer than 50% of patients present in these early stages, and they're presenting with advanced stage disease where they require chemotherapy along with surgical treatment strategies. Now, as you might be aware, June is also Bowel Cancer Awareness Month, and as a bowel cancer researcher, it'd be very remiss of me not to mention it and to encourage you all to be aware of the symptoms. If you have any symptoms, follow up with your GP. And of course, if you're in the age bracket where you are receiving those screening kits in the mail, please dig them out and do them. It might just save your life. So cancer is a complex and heterogeneous disease, and certainly colorectal cancer is no exception to this rule. We know that even when controlled for similar characteristics, that response to treatment is highly variable, and this is really driven by this inter- and intratumoral heterogeneity. And this can arise through different mechanisms, through genetic, epigenetic, and protein modifications. So in order to try and delineate these disease-resistant mechanisms and to try and identify more optimal treatment strategies for patients, we've worked on this collaboration for many years and established this living colorectal cancer bioresource. So this bioresource has been established by our team of scientists, surgeons, and oncologists over a number of years, and it has 3 main elements. So we have extensive patient data, which we can access for each of the patients who undergo surgery at Cabrini. We also collect tissue from patients undergoing resection, both in the form of fresh and fixed tissue. These fixed tissue specimens have been used for the construction of tissue microarrays, which now include over 1,000 colorectal cancer patients across all the different stages. And of course, we collect fresh tissue specimens, which we have been using to establish these patient-derived organoids. And we now have this really large bank of patient-derived organoids. So in our collection, we have over 100 primary tumouroid lines, or PDOs. Many of these PDOs have matched normal organoids, which we've derived from adjacent healthy tissue. And in some cases, we have lines from metastatic lesions matched to these primary and normal tissue samples from patients who have undergone synchronous resections. And we've established lines from these metastatic lesions in the lymph nodes, liver, peritoneum, and the omentum. And importantly, this collection reflects the wider colorectal cancer population. So looking back over this sort of collection of more than 100 different tumouroid lines, we've created this resource that's really reflective, as I mentioned, of the broader patient population. And we know this because all of the patients undergoing treatment for colorectal cancer at Cabrini are included in our extensive patient database. And this records over 350 different parameters for each treatment episode, and follows up our patients for up to 5 years. It also includes PDOs across all the stages, a good mix of genders, different tumour types, as well as lines harbouring key driver mutations and different molecular subtypes. Of course, now due to the breadth of this collection, we're fortunate to be in that position where we can focus on particular patient subtype, patient cohorts or particular subtypes of colorectal cancer. And this will really allow us now to further delineate this complex disease. And this bioresource forms the basis of a lot of our research questions that we're addressing in our lab. So just briefly, I wanted to touch on what are the advantages of using an organoid system over other models. So we know that more other commonly used disease models include cell lines and patient-derived xenografts. We know that cell lines cannot faithfully represent the original tumours. And whilst PDXs are a more complex model, they do take a long time to establish and success rate can be quite low. Of course, organoids are not without their limitations as well. They're certainly not without these limitations, they're a reductionist model based on epithelial cells alone. But advantages include that we're able to establish organoids from a really small amount of tissue. So we can establish an organoid line from a biopsy from a standard biopsy forceps. So that's a piece of tissue that's just a few millimeters in length. These organoids can be generated in a really short time frame. So as opposed to other cancer organoids, colorectal cancer organoids grow quite quickly. They are 3D cultures, which means not only do they maintain their cellular heterogeneity, but also their cellular hierarchy. And they mimic the features of the primary tissue from which they are derived. So as I mentioned, we've been, this collaboration between Cabrini and Monash has been ongoing for a number of years, and the majority of our patient samples have come from patients who are undergoing surgical resection for the treatment of colorectal cancer at Cabrini Hospital. So this is just a little about the process of how we're acquiring tissue, which might be helpful if you're looking to set up a similar type process in your own research. So our patients provide written informed consent for tissue and for access to their clinical data. We acquired tumour and normal colorectal tissue, and this is harvested directly from resection specimens in theatre by the surgeons themselves. So it doesn't go through pathology, it comes direct to us. These samples are rinsed in saline and placed into collection pots, and then we are notified that the samples are ready to collect. Our collection pot media is comprised of hibernate A, which is important for tissue preservation, and also a combination of antibiotics, which, as you can appreciate, is very important when we're looking at samples that are coming from the colon. Certainly the addition of primicin has reduced the amount of contamination that we do see coming from the patient samples themselves. The tissue is transported back to the laboratory where it is processed on the same or next day. And this is a bit of an overview of the process of establishing these organoids. So we take the tissue back to the lab and we trim and dissect the tissue. Some of that tissue can be fixed and frozen for further analysis of the primary tissue. The rest of that tissue can be chopped up into small pieces, can go through a series of wash steps followed by mechanical and chemical digestion before the cells are then seeded in matrigel in the bottom of these tissue culture plates. And the matrigel provides the scaffolding support for the organoids to grow in 3 dimensions. So this is the process that was sort of originally published many years ago by Hans Clevers in his laboratory. And over time, we've learned things and we've been able to refine it a little bit, particularly for small tissue samples or biopsies. We can actually skip the process of chemical and mechanical digestion and just plate small tissue fragments into the matrigel itself, and the organoids grow quite well. We've had a really good success rate using this methodology, and enables you to work with really small amounts of tissue. The other change we sort of made along the way, which has made this a bit more flexible, is that you can actually take these tissue samples and establish them at a later time if you wish. So you can cryopreserve the chopped tissue as you would for routine cryopreservation of cells, and this will allow you to establish the cultures at a later time. In our hands, we do find that these cultures tend to be a little slower to get going, to establish, as it compared to culturing directly from fresh tissue, But again, the success rate of growing organoids in this way is very high, in sort of over 90%, is in the ballpark of what we're seeing with our samples. So as I mentioned, we seed the cells into matrigel, and then we overlay them with our complete media. This is quite a complex media that contains essential growth factors and small molecules, and this mimics the in vivo microenvironment of the gut and supports the growth of these miniature tumours in the dish. I've just highlighted a couple of the key components here, and I just wanted to point out that whilst Wnt conditioned media or a Wnt surrogate is a requirement for culturing normal intestinal organoid cultures, colorectal cancer tumours commonly harbour mutations in genes associated with the Wnt pathway, and this is most commonly APC, and therefore the addition of exogenous Wnt is not required. So we capitalise on this, and we use these Wnt-free conditions to ensure selection and growth of tumour cells, that they're not outcompeted in culture by normal cells. So the full details of our culture conditions are available in our publications, but I just wanted to take this opportunity to point out that if you're looking to acquire these reagents, or you're looking for some technical assistance, we do have a Monash BDI organoid program, and this is now led by Dr. Terry Jardé, previously led by Professor Helen Abard. And if you're interested in using their services or getting some technical assistance, Dr. Genevieve Kerr, who's the program manager, is an excellent contact for that. So once we've gone through the process of establishing these organoids, what we really wanted to confirm then was that their organoids are actually a good model of colorectal cancer and the tissue from which they have been derived. So do they recapitulate the features of the tissue? So we've undertaken a number of different analyses to analyse our collection of organoids. We've performed whole exome sequencing and bulk RNA-seq, and we've been comparing the primary tumour specimens to their matched PDOs. We're also in the process of undertaking extensive proteomic analysis to further characterise the PDOs, but I won't be presenting any of that data today. In the initial stages, we just went about performing basic histological analysis on our organoids compared to our primary tissue. You can see in this panel that we've stained the primary tissue with H&E and the matched organoid in the second row there. And we see good correlation between these two. The CRC PDOs present with an epithelial architecture, which we would expect, and they have typical tumour-associated features. So we've also stained these organoids with markers that are used in the differential diagnosis of colorectal cancer, and this includes these three markers I've shown here: CDX2, CK7, and CK20. And the combination of these three, expression of these three markers is used to diagnose cancers of colorectal origin, and indeed our organoids do reflect what we see in the primary tissue of, from these samples. So we went on then to look at the whole exome sequencing data. We wanted to look at how well the PDOs reflect the mutational profiles of the primary tumour. And this plot here, this is bioinformatics work from our bioinformatician, Dr. Stuart Archer. This has been looking at the number of variants that were shared or seen only in the organoid, in the coral, only in the primary in the green, and then shared variants in the blue. And we do see a good maintenance and correlation between the organoid and the primary tumour. I just wanted to point out that these three patients on the end here are mismatch repair deficient patients. And these organoids clearly continue to acquire mutations at a very high rate in culture. And this is interesting, but unsurprising. We went further then to look at key driving mutations, and in this figure I've shown here the samples with the T representing the primary tumour and the O representing the matched PDO, and we do see a good correlation between these key driver mutations in the primary tissue versus the matched organoid. And this is over a number of different passages, and in one case up to passage 7, we do see that these organoids are stable over time. So we know now that the PDOs retain the key features of the primary tissue. I guess one of the big questions around developing these models is, can we actually utilize them to predict patient outcomes? So again, we've got this huge amount of clinical data that we can reflect on and compare what we're seeing in the dish in the laboratory to the clinical outcomes of patients recorded in our extensive database. And this is an example of some of the data that we're looking at in our metastatic patients, where we can look at the size of the tumours pre and post-treatment. So us and many other groups are obviously interested in examining the utility of PDOs for predicting patient outcome, but also for developing rational drug selection that could be used to sort of guide clinical treatment decisions. So importantly, colorectal cancer PDOs can be established in a very quick time frame, and therefore this sort of idea of taking a specimen from a patient and growing their organoid in a dish and testing with different drug combinations that will then ultimately be fed back to the clinician to help guide treatment decisions is actually really quite feasible in this setting. And colorectal cancer is sort of unique in that way, or in a very small club, I suppose, compared to other different cancer organoid lines that are slow to establish and can't be sort of expanded up into the numbers that we can see in our colorectal cancer organoids. So, of course, we went down this track where we were looking at whether we could mimic the prescribed treatment of prescribed treatments for each patient in their own matched patient-derived organoid. And this is just an example of a commonly used treatment combination of 5-FU and oxaliplatin, or referred to in the clinic as FOLFOX. And on the x-axis here is the increasing dose of 5-FU and oxaliplatin. And on the right, on the y-axis, the number of living cancer organoids. And so we can see over an increasing dose, the number of living organoids goes down, as you would expect. And we do see differential responses across the various PDOs. When we look at this data in the right panel, the area under the curve, the smaller the area under the curve, the more a patient is sensitive to that particular treatment. Again, we then went to compare multiple drug regimes for each PDO. We can do this on an individual basis, where we can compare 5-FU, 5-FU with oxaliplatin, and 5-FU with, and this is a combination of with SN38, which is a metabolically active version of irinotecan, and this mimics the FOLFIRI regime in the organoids. And we can see here that for this particular patient, the 5-FU and SN38 combination is more potent than the others. When we look at the response to, we can compare the response of primary tissue and metastatic PDOs, looking at whether these tumours that were taken from synchronous resections do respond similarly to treatment. So in this examples here, we've got primary tumours and metastatic tumours treated with the combination of 5-FU and oxaliplatin. And again, we see good correlation between the primary and the metastatic tumours in these examples. What we also see is good maintenance of morphological features between the primary tumour and the metastases in these examples. So over the past couple of years, there have been a large number of studies that have come out and reported on the ability to predict patient outcomes. And these have been with somewhat mixed results. So some report the ability to be able to predict patient outcome, but with limitations, including that it's only relevant for particular drug types, which makes sense depending on the mechanism of action. And each of these studies have really been based on really small sample numbers, particularly when we're looking at the correlation to actual patient outcomes. So what we have noticed too over time is that many of these rely on metabolic cell viability assays, which we know have a limited capacity to inform on cytotoxic and versus cytostatic effects. And I just wanted to highlight for you and show you an example of this as a bit of a cautionary tale, that in isolation, these metabolically metabolic assays may not be completely informative. So this is an example of an assay that I ran where I was looking at treatment with 5-FU, and we can see these examples here of organoid 139 compared to organoid 138. And on the graph here, they appear to respond quite similarly. What we do see, though, when we look down the microscope at these organoids is that the 1390, these organoids are very dead, and there's very few viable organoids remaining after treatment. Whereas the organoids on the right from patient 138 are very much alive. Whilst they're smaller than the control organoids, they're just static. They're not actually dead. And this is sort of an important point. We're looking at, if we can predict patient outcome, what are the assays we use and what are they actually telling us? Sort of as a reflection on sort of these mixed results we're seeing in the literature, you might think maybe organoids are just not a great model for predicting outcome, but perhaps we're just not utilizing them correctly. So how can we measure response more efficiently, and in what setting are they actually going to be predictive of outcome? So this is an example of some multiplex assays that we're running in our laboratory that we've developed over the last couple of years, where we're taking a sort of more broad approach to assessing cell viability outside of what we know from these metabolic assays. And what we can see here, that over an increasing concentration of drug, when we've stained for live and dead cells, that we have obviously in the lower end, more live cells, and as we increase, we get less calcine staining, less live cells there, and an increase in dead cells. And we're using these to develop these phenotypic assays where we're looking at measuring different aspects of the features of these organoids. And this is an example of some of the image analysis work that we've been working on, where this masking in the bottom panel is counting the number of live organoids in the top panel. And we can use this data to look at different measurements, including total organoid area or average organoid area, and have a different readout looking at how we might measure sensitivity to these drugs. Again, we see differential responses using this methodology. So what we're doing is developing these assays, looking at other parameters as well, including live dead cell counts, diameter, shape, and we're really benchmarking these criteria where we can get to a point then where we can determine which of these parameters or combination of parameters might better align with what we're seeing in terms of clinical outcomes. And of course, aside from utilising the PDOS for predicting outcomes, they're really useful models for other functional studies as well. And I just wanted to touch on some of that work in the last few minutes of my presentation, and ways that we're using these organoids in our preclinical work. So one of our projects has focused on characterising the revival stem cell population in colorectal cancer. So we and others have observed the reactivation of this fetal gene signature following treatment in colorectal cancer. And this study has focused on a gene that marks these cells, and this has correlated with treatment resistance. So these are a quiescent stem cell population that was originally identified within normal mouse intestine, and these are called revival stem cells. These cells are rare during homeostasis, but upon injury, they're enriched and they're capable of regenerating all of the other cell types of the stem cell, all the other cell types in the tissue. So we're interested in whether this subset of stem cells exists in tumours and whether they have a role in driving tumour relapse in patients. So firstly, we wanted to confirm whether revival stem cells actually exist in colorectal cancer, and this is some nice immuno work from a PhD student in our lab, Sarah Lifet. And these immunos show the expression of the revival stem cell marker clusterin in our colorectal cancer tissues. We then went on to examine the TCGA database, where we looked at the revival stem cell marker, which confirmed that clusterin, the expression of clusterin, is associated with poor patient outcomes. So this is data from a cohort of 200 patients, and those with higher clusterin expression have a decrease in overall survival and a higher risk of cancer relapse. So then we wanted to utilize our PDOs to determine if indeed there was a role for the revival stem cell in chemo resistance. So we performed drug testing in our PDOs, treating them with chemotherapeutic drug 5-FU, and we found that clusterin expression is increased following 5-FU treatment. And we also showed that basal clusterin expression correlated with chemo resistance. So we're now following up on these outcomes by further examining the role of clusterin in chemo resistance and disease relapse. And to do this, we are employing CRISPR technology, which allows us to generate reporter lines to help us answer these questions. And I just wanted to show you an example, not of the clustering line, but of a different reporter line that's been developed in our lab. So this work has really been pioneered in our lab by Dr. Horace Chan and Sarah Lavka, who've been working to label different cell types in our organoids for our functional studies. And this is just a really cool video where they've marked tubulin in our PDOs. We're also developing our high throughput imaging capabilities, which will be utilized for live cell imaging of these different experiments in an effort to delineate the key markers involved in driving chemo resistance. And with that, I'm going to end there. I just wanted to acknowledge the body of work that's really been contributed to by a large number of people over a large number of years. I couldn't possibly list them all on here, but I hope I've captured most of the people that have contributed to the work I've presented today. So a lot of this work, as I've shown, is from Horace and Sarah, the drug testing from my student Harrison. Stuart Archer has done a lot of the bioinformatics work, early work in this space by previous lab members, Terry and Jen. Of course, there's a huge team at Cabrini that also contributed to this study, including our team in the Department of Surgery, the surgeons, oncologists, pathologists, radiologists who all work together. I'd also like to acknowledge the platforms that help us do the work that we do here at Monash, and of course our funding bodies. And a special thank you to the patients who generously donate their tissue at a time where they're going through cancer treatment. It means a lot to us that they're willing to donate their tissue to our studies. And with that, I'm happy to take any questions.
All right, that was really great, Rebekah. Honestly, a tour de force. As someone who works in organoids, I understand that that's a lot of time and resources and input from many different facets of research, from the clinicians to the patients to the ethics to the clinical coordinators to everything. So it's a really big platform. You should be very proud.
Thank you.
I encourage people to ask questions, particularly the students, as it's a good opportunity to ask in a small forum. So while you're thinking about it and taking your microphones off mute, I'll start the ball rolling. I think just looking at it, some things came to mind which I was interested in. One is the heterogeneity. So I don't know how heterogeneous the original tumours are.
Mm-hmm.
These organoids become homogeneous. Do they maintain mutations? Is the transcriptional profile the same? And perhaps this is affecting your treatment response as well.
Yeah, I guess we're sort of learning about that as we go and sort of how they evolve over time in culture. They are quite stable in terms of, you know, things like driver mutations, but we do expect them, and they do accumulate different mutations over time. A lot of these are random and subclonal and not really going to affect the sort of the big picture, I guess. But we do see different clones expanding and dominating these cultures. So they do grow and change over time a little bit in terms of the cells that sort of grow out and become clonally sort of dominant. Whilst they don't become, while they're still heterogeneous, the ratios, I suppose, of the different clones does change as we culture them.
Have you ever looked, does that ever correlate with response?
We haven't done any sort of direct correlation probably, but I guess we're sort of more concerned about them maintaining sort of those key driver mutations that may have an impact on how we treat and the types of drugs we give them. But yeah, indeed, I guess if they're changing a lot over time in culture, it's I guess they may not correlate as well. I guess that's sort of interesting when we sort of reflect on those patients with the mismatch repair deficient, where they're changing quite a lot over time in culture. They're acquiring high rates of mutations. I guess we might expect that over time, they will be less reflective of the patient's tissue, and they may not be as stable and a good source of being able to predict outcomes for those patients.
Are there any questions from the audience? I can't see any hands. My next question was, I guess, about the contribution of the stroma. Have you ever attempted to, add anything, start to look at the CAFs, because obviously I think the stroma does play a big role in chemoresistance as well. Yeah,
absolutely. And it's certainly an area we're interested in, something we're looking at developing. We've got collaborative partners who are doing some of that work with us, where we're co-culturing the CAFs or fibroblasts with the organoids and sort of looking at how that might affect response. Obviously, those are going to have a huge impact on patient response. We know that the fibroblasts, they block T cells, they have a lot of interaction, the signaling, how it interacts with the epithelial cells is all going to play a big part in that. So I guess, we're sort of developing these models. We're also trying to develop them back towards a more physiologically relevant kind of model where we're adding back in some of these other elements. When we can add in fibroblasts, we can add back in T cells, and then we can sort of look at a more big picture of how we might use these in a more physiological manner, and maybe then improve our results in terms of correlating back to patient outcomes.
Did Karen Harris have a question?
You saw me take myself off mute for a moment. Hi, I'm Karen from the MPCCC. I did have a question, actually. You talked about the 350 parameters that you kind of look at, and that your samples are representative of the wider population. And I was just wondering, but that it's also just from Cabrini. And so I was wondering if there's samples that come from, Victoria-wide across other institutes, if this is a program that others are doing as well. And also about whether you could tell us a little bit more, sorry if I missed this throughout the talk, but tell us a little bit more about what those parameters are that tell you that you're getting a good representation.
Yeah, of course. Yeah, so that is, I guess, a bit of a limitation of our collection. We are collecting from patients at Cabrini primarily. We do get some of our patients through some of the clinics, local clinics, who are recruiting from patients with suspected rectal cancer. So we are trying to broaden out our horizons a little bit. We know there are lots of people sort of across Australia who are working on these similar types of collections, and we've collaborated with a group in Adelaide and other groups in Melbourne as well to try and sort of capture a more broader spectrum, I suppose, of patients, to try and sort of more broadly capture. I haven't spoken about that work, but sort of what we're, in terms of that collection and sort of looking at comparing, I guess, across different sites, how robust this system might be. In terms of the parameters we're collecting, we're really looking for trying to use this information to make sure we're covering different tumour types, different primary locations, patients with, of course, all different staging, from adenomas through to stage 4 disease. Obviously, sort of, basic information like patient age. We have a huge spectrum of patients, and we're really getting a lot more patients coming through in that younger category as well. And we follow these patients. So we're looking at how they track over time. We follow them up for up to 5 years. We can look at how they're treated. And we're also sort of moving into, so I'm not sure if I mentioned, but a lot of our collection really is based around these treatment naive patients. So patients who haven't had systemic therapy, but to sort of evolve our collection and sort of move into asking some of these more complex questions, we're also looking at patients who have been treated. And so we're looking at trying to expand our collection to sort of look at those sort of the different aspects of what treatment might do to different clones, and looking at how that might affect their response to treatment as well. So we're really trying to capture sort of a real, like, sort of as much as we can, I guess, in that way across different cohorts of patients. Does that answer your question?
Yeah, no, that's great. Thank you.
That's great. Are there any further questions?
I had another question if no one wants
to
ask anything. I just wanted to ask about your assays, where you talked about, you know, being able to assess the impact of different drug combinations in real time. I suppose there's two questions. One is about what realistically, what are the timelines on that, and could you see it really happening, anytime in the future? And another question was, would you see that as being something that's complementary to genetic testing and the panels that are being used now?
Yeah, of course. So in terms of time frame, colorectal organoids do grow really quickly. So we can establish organoids. It varies, obviously, patient to patient. Every patient's a little bit different, but we can establish organoids sort of within a couple of weeks. We can have a line going. So by the time a patient has surgery and then they're recovering from their treatment, but before they have chemotherapy, we could establish these organoids in numbers sufficient to run these drug screens. And the drug screens would be an additional sort of couple of weeks, I suppose, is sort of where we're sort of heading towards. So in a period of sort of 4 to 6 weeks, depending on the patient tissue, we could take this tissue, establish the organoid, and have this information back to the clinic. And of course, yes, this would be part of the picture. I guess we can tell them what we learn, but this has to also be taken in consideration with other clinical factors that we know might impact a patient's ability to respond to different drugs, and I guess to their ability to tolerate different drugs. So we see this becoming part of the picture that would be complemented by genetic testing and other important factors that can contribute to a way a patient might respond to treatment.
Yep, I think I just had a similar thought actually about that as well, like how easy is it to get into the clinic? I mean, it might be easy for approved drugs, but obviously for anything experimental, you're going to need to advance to more larger clinical trials.
Yeah, exactly. So I guess we're sort of focusing our interests on drugs that are that have safety profiles and have maybe uses in other cancers, or are currently in clinical trials, so that I guess it would make what we're doing more translatable more quickly, with the idea then that we could look at novel treatments as well, but obviously with a longer time frame to getting them into the clinic.
That's great. So if there aren't any further questions, we will conclude here. I just wanted to thank Rebekah very much for giving her talk. It was a really nice snapshot into some of the work they're doing. I'll hand over to Hannah, who's going to introduce our speaker for next week, I believe, or in 2 weeks' time, I think.
Hi. Yes, so in a couple of weeks' time, we have a tech talk, and you can go onto our website, MPCCC, and if you go to our events section, there is a registration button there. You can log on, and that will go into your calendar. So it's Dr. Christy Ying, and she'll be talking about gastric cancer. Yeah, thank you so much to Rebekah and to Natalie for today. That was great talk, and follow us on Twitter and LinkedIn to keep up to date with all of our talks. But yeah, that was great. Thank you very much, everyone.
Thanks.
Thank you so much.
See ya. Bye.
Thank you. Bye.
Bye.